This video aims to educate you on Zopiclone. We will look into what type of drug it is, what it's used for, how it was discovered and developed into a drug, its traditional synthetic route, then the main side effects, and finally the mechanism of action of the drug. Xavier is a pharmacy student. He's very stressed, and it dawns on him that he's not been able to sleep for a few nights now, and he thinks this is a problem, so he decides to go and see his GP. Insomnia is essentially sleeplessness. It can include difficulty falling asleep or staying asleep for long enough that you feel refreshed the next morning. As adults, we're required to get between 7 and 9 hours of sleep per night. And if we do not get this, it can cause detrimental side effects to our body. There are 3 main types of insomnia, the first being transient insomnia. This is caused by things such as noise disturbances, shift work, or even jet lag. These things mess up your body's natural body clock and can sometimes lead to difficulty falling asleep. In the case of transient insomnia, a very short-term treatment with a hypnotic such as Zopiclone should be used to help the condition. The second type of insomnia is chronic insomnia. This is usually drug and alcohol induced, but it can also arise from depression or anxiety. Hypnotics would not usually be used to treat this type of insomnia, as it is caused by an underlying issue, and it would therefore benefit the patient to identify the issue, treat the issue, and eventually the insomnia will be resolved as well. The last type of insomnia is short-term insomnia. It's probably the most common type. It's insomnia induced by stress, which is what Xavier is suffering from, emotional events such as death or trauma, and can also be an effect of a serious medical illness. In this case, Zopiclone would be used for up to 4 weeks. Xavier receives his prescription of Zopiclone from the doctor, but he's still eager to know more about the drug. So he goes home and researches. In 1964, Roane Poulenc carried out a broad random screening program aimed at the search of anxiolytics. From their 3,000 lead compounds screened, they discovered Zopiclone, a cyclopyrrolone hypnotic. Zopiclone was the first compound developed which is chemically unrelated to benzodiazepines, yet binds of high affinity to benzodiazepine receptors. Random screening starts with a large screening library. The compound repository is built from the historical collections, natural products, and combinatorial synthesis. In the initial high-throughput in vitro assay, primary hits are identified. The random screening assay is used as a filter to identify the promising hits, which are used to justify further analysis for lead finding. Normally, all the data can be used in subsequent analysis. Functional tests can monitor interactions with different binding sites. In the review process, the initial paradigm is developed to design combinational libraries and to populate the screening library on the basis of properties that have proven to be associated with successful therapeutics, especially physiochemical and ADME properties. Between 1987 and 1989, the different protein subunits that make up the receptor for the inhibitory neurotransmitter gamma-aminobutyric acid, GABA, were identified as alpha, beta, gamma, and delta. These relate to the specificity of drug receptor binding. Zopiclone specifically binds to the alpha subunits, as we'll see later on in the video. Zopiclone has been sold in France since the late 1980s. In 1999, Avantis Pharmaceuticals, which is now Sanofi, rightly owned the preclinical, clinical, and post-marketing data package for Zopiclone, its isomers, and its metabolites. In 2004, US FDA approved S-Zopiclone under the brand name Lunesta for the treatment of insomnia. This was after around 24 clinical trials that included more than 2,700 adults and elderly subjects, as well as data from more than 60 preclinical studies. In 2005, S-Zopiclone, the active S-enantiomer of Zopiclone, arrived in the market. It's supposed to have equivalent efficacy with racemic Zopiclone and possibly fewer side effects than that of Zopiclone. There are lots of methods in synthesizing Zopiclone, particularly in the last stage of synthesis. However, in this video, we're going to talk about the traditional synthetic route. The starting material for the synthesis is pyrazine 2,3-dicarboxylic acid. This molecule is dehydrated in the presence of acetic anhydride and toluene to produce pyrazine anhydride. After this, condensation occurs by combining pyrazine anhydride with 2-amino 5-chlorpyridine through nucleophilic acyl substitution to give an amide with a yield of 92%. The process is undergone in acetonitrile and in reflux temperatures. Next, an imide is formed by the cyclization of amide produced in the previous step with the aid of thionyl chloride at reflux temperatures with a yield of 77%. The fourth part of synthesis is completed by the monoreduction of carbonyl oxygen of the imide to a hydroxyl group with the product yield of 64%. This route is mediated by potassium borohydride, dioxane, and water. The final step of the synthesis involves the condensation of the intermediate produced from the last step through the acyl substitution in the presence of sodium hydride, dimethylformamide, and CCMP at -10 degrees to eventually give racemic Zopiclone. The yield at this step is measured at 43%. Once finished with his research, Xavier takes his prescription to pharmacy and is told about the side effects and how and when to take Zopiclone. The pharmacist tells Xavier the dose he should take is one 7.5mg tablet at bedtime. He then goes on to say that Xavier could experience a bitter or metallic taste in the mouth, dry mouth, and daytime sleepiness. And if he feels drowsy in the morning, he should not drive or operate heavy machinery. These side effects are common, but not everyone will experience them. These side effects occur because Zopiclone binds to the GABA receptor at the benzodiazepine binding site at the positions alpha 1, alpha 2, alpha 3 and alpha 5 receptors as full agonists, causing an enhancement of the inhibitory actions. Research shows that the alpha 1 subunit is associated with sedation, the alpha 2 and 3 subunits are associated with anxiety, and the alpha 5 subunit is associated with temporal and spatial memory. In this case, alpha 1 subunit receptor is the intended receptor, whereas alpha 2, 3 and alpha 5 are the unintended receptors. However, even though the alpha 1 subunit is the correct receptor, when taking an overdose of Zopiclone, it may present as excessive sedation and depressed respiratory function that may progress through coma and possibly death. There is also a relationship between high GABA levels and depression, which makes sense given that GABA typically promotes a calm, relaxed mood, but being too calm can turn into indifference and apathy, hence why a patient may feel dizzy, lightheaded, or faint. The human body may also become tolerant to Zopiclone, which means higher doses would be needed to achieve the same effect. For this reason, Zopiclone should be taken with caution, as directed by the prescriber, and at the recommended dosage. It should also be weaned off slowly, rather than abruptly stopped. Xavier takes his prescription home and at bedtime takes his correct dose. We can now start to understand the mechanism of action of Zopiclone and how it is used to treat insomnia by looking at its structure and activity. As you can see, Zopiclone is a small molecule with a log P of 0.8, 6 hydrogen acceptors and no hydrogen donors. It has a molecular weight of 308.8. This means that Zopiclone fits all 4 of Lipinski's rules, and for this reason it is orally available, most commonly as a tablet. Zopiclone is a cyclopyrrolone, a family of hypnotic and anxiolytic non-benzodiazepine drugs. These two molecules are also cyclopyrrolones. They have similar structures to Zopiclone. They also have a sedative effect like Zopiclone. Since the pharmacophore for Zopiclone hasn't been established, we can suggest what it may be according to these two drugs. It displays ionic interactions, pi-pi stacking, and hydrogen bonding. These would be the factors considered when looking at its activity on the benzodiazepine receptors. There is no precise mechanism of action known for Zopiclone, but we can establish that its action is closely related to its ability to bind to the benzodiazepine receptor complex. The cyclopyrrolone in Zopiclone recognises a site on the benzodiazepine receptor complex in the GABA alpha receptor and binds to it, causing an allosteric change. Zopiclone has improved to become S-Zopiclone over the years, which is the S-enantiomer of Zopiclone. The S-enantiomer reduces the residual side effects of Zopiclone but has a shorter half-life. The structure-activity relationship is not completely established for Zopiclone, but it is known that S-Zopiclone recognises a distinct site in the GABA receptor complex allosteric to the recognition site for benzodiazepines. Distribution and specificity of the site of cyclopyrrolone is similar to those of benzodiazepine, suggesting that cyclopyrrolin binding site resides on the benzodiazepine site of the GABA complex. Now let's focus on the GABA receptor. There are neurons in the central nervous system that use the amino acid GABA as a neurotransmitter, which is unique among amino acids in that it is manufactured almost exclusively in the brain and spinal cord. GABA acts as a natural tranquilizer, mitigating the stress response and lowering anxiety levels, as well as slowing down functions of the central nervous system. This means that when these neurons fire, they secrete GABA into their synapses. When GABA receptors are stimulated by the presence of GABA, they typically inhibit the postsynaptic neuron from firing. When GABA alpha binds to the receptor, it allows the flow of the chloride ions through a special channel in the membrane. This flow causes inhibition of the neuron firing. In addition to a site for GABA, the GABA receptor has a site for the Zopiclone molecule. When the molecules are present on the GABA receptor, they increase the efficacy of GABA to bind to the receptor. However, Zopiclone binds with roughly equal affinity to GABA alpha receptors containing different alpha subunits. It is therefore not selective as to which GABA-dependent behavioural effects, such as sedation and anxiolytic effects, occur. When the receptor is activated, this chloride channel opens, causing negatively charged chloride ions to enter the postsynaptic cell. This makes the neurons less responsive to stimulation by making the membrane potential more negative or hyperpolarized. The widespread distribution of GABA receptors suggests that the brain is generally in a state of inhibition. Xavier has now woken up from a perfect, undisturbed night's sleep and is ready to continue with his university life without the added stress of being unable to sleep properly.